Welcome to the Deep Dive, where we break down your sources and really get into the good stuff. Today, we're diving into bacterial gene regulation, and you've sent in some textbook excerpts on it. Get ready to see how these tiny organisms fine-tune their genes with amazing precision.
We'll be covering operons, inducers, repressors, and we'll even get into the details of the lac and trap operons in E. coli.
Okay, let's jump right in. We think of bacteria as simple, but their efficiency is, well, it's anything BUT simple. Take E. coli, for example. It's evolved these incredibly efficient ways to control which genes are turned on or off.
That's right. It all comes down to adapting to their environment and using resources wisely. Think of it like this, a single-celled organism constantly adjusting its inner workings to survive. That's what we're talking about here.
It's more than just flipping a switch for enzyme production, though, right? They're coordinating DNA replication, repair, even cell division, all while reacting to the environment.
Exactly. And to manage this, they use two types of enzymes, inducible and constitutive. Inducible enzymes are only produced when a specific target molecule is present. Like when lactose is around, E. coli will make enzymes to break it down. But if there's no lactose, well, it doesn't waste energy making those enzymes.
So it's like an on-demand system for specific tasks. That makes the constitutive enzymes the always-on workers, regardless of what's happening around them.
You got it. But the big question is, how do bacteria actually control this enzyme production? That's where negative and positive control mechanisms come in.
Negative control sounds like a default on setting until there's a signal to stop. So positive control would then be waiting for that go signal.
Exactly. A perfect example of both these mechanisms is the lac operon, a group of genes in E. coli that helps it use lactose. It's a classic example of bacterial gene regulation, and it really shows how negative and positive control work together.
So let's break down negative control in the lac operon first. When there's no lactose, a repressor protein binds to the operon's operator region and blocks transcription.
Right, it's like a roadblock stopping those enzymes needed to break down lactose from being made. No lactose, no need for the enzymes, so the system is off by default.
But when lactose shows up, it binds to the repressor and changes its shape. This causes the repressor to detach from the operator, opening up the road for transcription.
And with the roadblock gone, RNA polymerase can transcribe those genes, and the cell can start making those lactose-digesting enzymes.
Scientists have even found mutations that affect this whole process, which supports this model, right?
Yeah, they found specific mutations in the repressor gene that stop it from binding to the operator. This leads to constant expression of those lac operon genes even when there's no lactose around.
Wow, that's really interesting. But the lac operon isn't only regulated by negative control, it also uses positive control through the catabolite activator protein, or CAP. I've heard of CAP before, but I'm a little fuzzy on how it works. Doesn't it use a molecule called CAMP?
You're right, it does. When glucose, E. coli's preferred energy source, is low, CMP levels increase and bind to CAP. This CAMP-CAP complex then binds near the lac operon promoter, which makes it easier for RNA polymerase to bind and start transcription. So basically, CAP boosts the lac operon signal when glucose is scarce.
So CAP helps E. coli use glucose first, but when glucose runs low, it gives the lactose processing system a boost. But wouldn't that be a problem in environments where there's tons of lactose but no glucose? Wouldn't always prioritizing glucose be a disadvantage in those cases?
That's a great point, and it really shows the balancing act bacteria have to deal with. While prioritizing glucose is usually a good thing, there are definitely situations where being able to use other sugars, like lactose, is crucial for survival. We'll dig deeper into this trade-off a bit later. For now, let's move on to another key operon, the trap operon, which is all about making the amino acid tryptophan.
The trap operon is different from the lac operon because it's repressible, right? So it's usually on, producing those tryptophan-making enzymes.
Exactly. But when there's a lot of tryptophan already, the cell doesn't want to waste energy making more. In these cases, tryptophan actually acts as a core pressor, binding to the trap repressor protein.
So unlike the lac operon, which uses an external inducer like lactose, the trap operon uses its own product to regulate itself. That makes sense for efficiency.
Absolutely. This tryptophan repressor complex then binds to the operator and stops transcription. It's a classic feedback loop, making sure the cell only makes tryptophan when it actually needs it.
Wow, this is all pretty complex. The lac operon uses both negative and positive control, and then the trap operon has this feedback loop. Bacteria really are masters of gene regulation.
Well, they definitely are. And we've only scratched the surface here. The trap operon has another trick up its sleeve, attenuation. But we'll get into that in the next part of our deep dive.
This is already blowing my mind. So much for bacteria being simple. We'll be back soon to unpack this attenuation thing in the trap operon. Stay with us.
Welcome back to the deep dive. We're digging deeper into bacterial gene regulation, and last time we were talking about the trap operon, that repressible system using a feedback loop to control tryptophan production.
Yeah, we discussed how excess tryptophan acts as a core pressor, binding to that repressor protein and basically shutting the operon down.
Right, but you also mentioned something called attenuation, which adds another layer of control on top of that. So what is attenuation exactly, and how does it actually work?
Well, attenuation is this pretty cool mechanism that lets bacteria fine-tune gene expression based on how much tryptophan is available at that exact moment. What's really interesting is that it works at the mRNA level, the molecule that carries the genetic code from the DNA to those ribosomes where proteins are made.
Oh, so it's not directly controlling if the gene is transcribed, but rather how that message, like the transcript itself, is handled. You got it. It uses a leader sequence inside the mRNA, which is a stretch of nucleotides that comes before the actual genes that code for those tryptophan synthesizing enzymes. This leader sequence can fold into different shapes, secondary structures they call them, depending on the concentration of tryptophan in the cell.
So the mRNA is like a sensor for tryptophan levels.
Exactly. Imagine the leader sequence is like a flexible ribbon. When tryptophan levels are high, it folds into this specific hairpin structure that basically looks like a termination signal for RNA polymerase, the enzyme that transcribes DNA into RNA.
So is it basically like when there's enough tryptophan, the mRNA tells RNA polymerase, hey, you can stop making the transcript now?
Yeah, that's the main idea.
Yeah.
But when tryptophan levels are low, the leader sequence folds into a different structure, one that doesn't look like a termination signal. And in that case, RNA polymerase just keeps transcribing those genes for tryptophan synthesis.
So it's like the mRNA is a switch, flipping between stop and go based on how much tryptophan is around. It's pretty wild how something as simple as mRNA folding can have such a big impact on gene expression.
It really shows how clever evolution can be. But it gets even more interesting when we look at how the choice between those different mRNA structures is made. This is where the ribosome, the protein synthesis machinery of the cell, comes in.
Wait, the ribosome? I thought we were talking about transcription, not translation.
Well, attenuation is this great example of how transcription and translation are actually linked in bacteria. Remember, during translation, the ribosome moves along the mRNA, reading those codons and adding the matching amino acids to the growing protein chain.
Right, so how does that connect to attenuation?
Okay, so within the leader sequence of that operon mRNA, there are two codons that specify the amino acid tryptophan. Now, when tryptophan levels are high, there's a lot of charged tryptophan tRNA available. This means the ribosome can easily translate those tryptophan codons and move along the leader sequence without any problems.
And this smooth movement of the ribosome changes how the mRNA folds.
Exactly. When the ribosome zips through those tryptophan codons, it lets a certain part of the leader sequence, we'll call it region 3, pair up with another region, region 4. And this pairing creates that terminator hairpin we talked about, the one that tells RNA polymerase to stop transcription.
So the ribosome, by translating those tryptophan codons quickly, is indirectly helping to form that termination signal. But what happens when tryptophan is scarce?
When tryptophan is low, there's not much charged tryptophan tRNA around. So the ribosome, when it gets to those tryptophan codons, it stalls, like it's waiting for the right tRNA to show up.
And the stalling affects the mRNA folding.
Absolutely. Because the ribosome is stuck, region 3 can't pair up with region 4. Instead, a different pairing happens. Region 2 pairs with region 3. And this different pairing actually stops the terminator hairpin from forming, which means RNA polymerase can just keep transcribing those operon genes.
Whoa, so the ribosome, just by pausing at those tryptophan codons, is indirectly controlling the mRNA structure and deciding whether transcription continues or stops. It's like this back and forth between transcription and translation, with the ribosome acting like a sensor for how much tryptophan is there.
Yeah, that's a great way to put it. Attenuation is a perfect example of how bacteria have evolved these complex ways to fine-tune gene expression based on their constantly changing environment.
This is all incredibly intricate. Are there any other operons that use attenuation, or is it just the trip operon?
Attenuation is mostly seen in operons that are involved in making amino acids, like the trep operon. It's a really elegant solution for systems where the final product of the pathway can directly influence its own production,
So it's like this built-in feedback mechanism at the mRNA level. It's amazing how bacteria can fit so much complexity into such tiny systems.
It really is. And studying these bacterial systems has actually been super important for understanding basic genetic principles. What we've learned from studying operons like lac and trap has helped us understand gene regulation in more complex organisms too, even humans.
That's amazing. But I'm curious, how does this knowledge actually apply to those more complex organisms that don't have operons in the traditional sense?
Well, while eukaryotes don't have operons, they do have their own sophisticated mechanisms for regulating gene expression. One of those is alternative splicing, a process where different combinations of coding sequences within a gene can be included or excluded in the final mRNA.
So it's kind of like choosing different parts of a recipe to make slightly different versions of the same dish.
Yeah, that's a great analogy. And this alternative splicing can be regulated by a bunch of factors, like the availability of certain proteins and the environment inside the cell.
So in a way, it's similar to attenuation, where the availability of specific molecules can impact how the genetic information is processed.
Exactly. Well, the specific mechanisms are different. The underlying principle of fine-tuning gene expression based on what the cell needs is something that's shared across all life forms.
It's incredible to see how these seemingly simple bacterial systems have actually revealed these fundamental principles of gene regulation that apply to all life.
It is pretty remarkable.
Yeah.
And as we keep exploring all the details of gene regulation, we're constantly reminded that some of the most important lessons can come from the tiniest creatures.
And we're back for the last part of our bacterial gene regulation deep dive. I gotta say, I'm still thinking about that attenuation mechanism in the trap operon. It's wild how dynamic mRNA can be.
Yeah, it really is amazing how much control those little molecules have. It goes back to what we've been talking about, how bacteria, even though they're tiny, are super sophisticated in how they regulate their genes.
We've seen how they use repressors and inducers and even like mRNA gymnastics to respond to their environment and not waste resources. It's like they figured out efficiency on a whole other level.
Exactly. And the cool thing is, these bacterial systems aren't just isolated examples. They've helped us understand some really basic principles of gene regulation that go way beyond just the microbial world.
So the stuff we've learned from bacteria can actually help us understand gene regulation in more complex organisms.
Oh, yeah, for sure. The ideas of operons and repressors and activators, all that stuff was first discovered in bacteria. And it turns out they're like universal themes in how genes are regulated across all life.
Even attenuation, which seems so specific to bacteria, has parallels in eukaryotic systems, right?
You're thinking of alternative splicing, where different parts of a gene's code can be included or left out of that final mRNA. It's like, creating different versions of a protein from the same blueprint, depending on what the cell needs at that time.
So even though eukaryotes don't have operons exactly, they still have all these complicated ways of fine-tuning gene expression.
Exactly. And this understanding, which we got from studying bacteria, has really changed how we think about human health and diseases.
Because a lot of genetic disorders are caused by problems with gene regulation, right?
Right. Like, if there are mutations in genes that code for those transcription factors or regulatory elements, it can mess up the whole system. That can lead to all sorts of problems that show up as diseases.
And because we've studied those simple bacterial models, we've learned more about how those problems happen and how we might be able to fix them.
It's like having a basic map to help us understand the more complicated machine of eukaryotic gene regulation.
It's pretty amazing to think that these tiny organisms that we often think of as simple or even harmful have actually taught us so much about life itself.
We really have.
Yeah.
And as we learn more about gene regulation, we keep seeing how much we can learn from even the smallest creatures.
This whole deep dive has definitely changed how I think about bacteria. They're not just simple cells, they're like masters of genetic control, constantly adapting and changing to survive.
And who knows what else we'll learn from them. There's still so much to discover about bacterial gene regulation.
This deep dive has definitely made me even more curious. Like, I'm wondering about how we could use this knowledge. Could we maybe engineer bacterial gene regulatory systems to make biosensors for environmental pollutants?
That's a great question and a perfect example of how we can use this basic knowledge. There's a lot of potential there, and people are already doing research on it. Imagine using bacteria to detect toxins in water or even in our bodies.
The possibilities are pretty incredible. It's amazing that something as small as a bacterium could be the key to solving some of our biggest problems.
It just goes to show that we can still learn so much from the natural world, even from the organisms we might not even notice.
Well, I think it's safe to say this deep dive into bacterial gene regulation has been a success. We've learned about the amazing ways these tiny organisms control their genes, and I think we both have a new appreciation for how complex and sophisticated they are.
Yeah, definitely. And I hope this has sparked some curiosity in you, our listeners, to keep learning about biology and genetics.
Because there's always more to discover, and sometimes the smallest organisms can hold the biggest surprises. Until next time, keep on diving deep.